interested in trying to measure the evolutionary dynamics in human tumours, and particularly in solid tumours. And so I want to tell you about some work that we've been doing with Christina that's been principally led by Mark in my lab, who's a very talented PhD student, with Andreas Otteriva, who you've heard about at the ICR in London, and it's funded by Cancer Research UK and by Wellcome. And so we're interested in what the evolutionary dynamics are and how these evolutionary dynamics shape the cancer genome that we observe. And so I think there's three components to the evolutionary dynamics, and we hear a lot about this one on the right. We hear a lot about the impact of selection and about driver genes and how these interesting bits of evolutionary biology for selective events shape what we see in the genome. But we actually hear a bit less, I think, about the role of chance, about the role of genetic drift and non-selected random dynamics, which can have a very large effect on the cancer genome too. And that's something we've tried to focus on a lot, to understand the effects of these random chance genetic drift events and random mutation on shaping the cancer genome. So the talk I'm going to give is very much a genetics talk, but it's not to say that I think the microenvironment is not important. Clearly, the microenvironment has a key influence on all of these aspects. We heard from Nikki say that how microenvironmental influences such as mutagen exposures can affect the mutation rate and change the chance of different random mutations happening. We heard a little bit from Christina about how the histological structure affects the neutral drift dynamics that can happen in tissues. In the colon, for example, we have these crypts, and those crypts contain small numbers of stem cells and strongly constrain the patterns of evolution that can happen within those histological structures. So these microenvironmental influences have strong effects there, and they clearly, as we've just heard, have strong effects on the consequences and the ability of selection to act. So the goal of what we're trying to do is to read out these underlying causes of the evolutionary dynamics simply from the cancer genome itself. We know that the microenvironment has a strong influence, but we don't necessarily need to measure it to see the consequences of these selective forces at play, say, that are driven by the microenvironment. So we're interested in measuring what the evolutionary dynamics are, but glibly, this is the problem, right? So this is a problem that we have to deal with. The only thing we ever have to look at is a big, ugly cancer. There's a whole occult process of evolution that happens that we never get to observe for obvious reasons, and all that we ever have is the tumour, which is resected and ends up on our specimen table at the end. So the challenge is for us to try and look back in time and understand these secret evolutionary dynamics that led to the production of this tumour, but to try and understand it only by being able to measure at this very end point in time, only by sampling this tumour when it's ended up on the resection table. And as many people have said today, and we started with a beautiful talk from Dan Landau this morning, our approach is to use somatic mutations to trace clonal lineages in tumours to try and infer this occult evolutionary history. So we have this kind of model of tumour growth that begins at some point in time after a cell has some critical has acquired a critical set of alterations that are sufficient to give it a growth advantage in its current microenvironment, and that microenvironment may change during tumour progression. And then that instigates cell growth. So over a period of time, we'll have a number of cell doublings that go on that lead to the growth of a tumour. And we know that in cancer, the mutation rate is quite high, and the chances are that every time a cell divides, there's a new mutation somewhere in the genome. And those mutations are, of course, inherited, so they give a unique lineage label to every single cell in that tumour. So if we want to know the history of a particular clone in a tumour, it suffices just to find the mutation that marks that clone. Say, for example, we may be interested in the evolutionary history of this cell and the clone that it generates. We can just use the mutation that uniquely marks that cell to trace its history. So in this cartoon example, that's this blue hexagon. So these somatic mutations are a natural lineage tracing mechanism that the tumour, in some sense, fortuitously creates for us to study. And an implicit, most of these mutations that accrue over time do nothing. They're just random passenger mutations that have no effect on fitness. And it's those passenger mutations that are really useful to study because they're the ones that reveal the evolutionary history of the tumour. And so we read these mutations through next-generation sequencing. frequency spectrums or variant allele frequency histograms that Christina explained, and these are the data that we work with to try and understand what these evolutionary dynamics are. So I'm sure that most people in the room are very familiar with these plots, but just in case, on the x-axis we have the measured allelic frequency of a mutation, and this is a histogram, so on the y-axis we have the number of mutations that are measured at that frequency. And this is a very typical cancer genome plot. It often you see a peak of mutations at high frequency in the tumour. These are the mutations which are in every single cancer cell. This is this big set of mutations denoted by this letter C in my cartoon. These are the clonal mutations that are present in that founder tumour cell. And so the frequency should be at 0.5, right? Human cells are diploid, but it's not. It's compressed down to a lower frequency by the stromal cells in the tumour, which are genetically wild type, and geneticists sometimes rather rudely call them contaminating normal cells, when actually they're very integral to tumour biology. But nevertheless, they do dilute the signal of genetics that you see. And so the spread is because of the limited depth of sequencing, so we inaccurately measure the allele frequency. So this is why we don't just see a spike. And then all of the mutations at lower frequency are these mutations which have accrued subsequently post that first tumour cell during the growth of the tumour that we have. All the heterogeneous mutations in the tumour, all of the subclonal ones. And it's this pattern of mutations here, this pattern of subclonal mutations in this tumour, this pattern of heterogeneity, which we try and read out to understand how the tumour's grown. And so it's quite easy to get these data now, and we've seen lots and lots of examples of these data. And our question is, how do these allele frequency histograms tell us about the evolutionary dynamics of tumour growth? You know, what can we learn from these diagrams about how the tumour grew? And so our approach, so I'm a mathematician by training, is to make a mathematical model of parameters in our mathematical model of tumour growth explain the data that we see, and can we therefore make inferences about what the actual parameters of evolution are? So this is a multi-scale model inherently. It starts with cells, which are one length scale, and then we model the DNA, which is of course a much smaller length scale. And so we're linking across length scales in this multi-scale model, and we use this multi-scale model to understand the genetic data that we observe. So it's an approach we sort of nickname physical bioinformatics, because it combines mechanistic models of cell division and growth together with the genetic data, and is a different way of thinking about interpreting the genetic data. So the first question we wanted to ask was, well, what happens if nothing happens? We've seen lots of heterogeneity in the tumour. How much heterogeneity should we expect if there's no clonal selection at all, if there's no interesting evolution going on? And so this is a well-described question in evolutionary biology, and it's neutral theory. So neutral theory is a well-developed theory, null hypothesis for molecular evolution. So we wanted to know, well, what does neutral evolution look like in a growing tumour population? Can we see a signature of neutral evolution in the tumour genome? So to ask this question, we first made a mathematical model of neutral evolution in a growing tumour. So in neutral evolution, we have growth, so we have a branching process with exponential growth. There's no clonal selection, so all of the lineages within this model behave the same as one another. The chance of any individual lineage having a number of offspring is the same for all lineages, and we have a constant mutation rate with a Poisson distributed number of mutations at each division. And so the kind of intuition you can imagine about what neutral evolution looks like in these variant allele frequency histograms comes from just drawing the picture. So we start with our first tumour cell with its complement of mutations that are sufficient to drive tumour growth. And then this cell starts to divide, and each time it divides, those daughter cells get some new mutations that uniquely mark their lineage. And so we can imagine, so after this first round of division, there's now, say, 2 new mutations, one in each of these 2 new cancer cells, and each of them are at frequency a half. They're in one or other of the tumour cells. So we get, we can draw a graph of this and start to look at the predicted variant allele frequency spectrum under this neutral model. And so the next round of divisions say there's more new mutations. There's now 4 new mutations in our population. They're all at frequency a quarter. and so on and so on. The next round of division, 8 new mutations, all at frequency an eighth, and so on. And you can see you get a characteristic curve under this neutral model of ever more mutations at ever lower frequency as the tumour grows. And so we can, because this is a mathematical modelling talk, you can of course derive an expression for exactly what the site frequency spectrum, the variant allele frequency histogram should look like under a neutral model. So I'll just whiz you through the maths for the mathematicians in the audience. So we're interested in how the total number of mutations changes as a function of time, and that's just the population size times the mutation rate times the rate at which the population grows. So this lambda parameter just scales time to divisions, and the pi parameter is the ploidy of the cancer cell, so changes the population size into the number of alleles in the population. The frequency of a mutation doesn't really change over time. It's determined just by the size of the tumour population at the time which the new mutation arises. So with this in mind, we can we can substitute time for frequency back into this equation and do the simplification and get this useful formula which tells us the expected variant allele frequency spectrum, which is time independent. So this distribution is constant in time. And it has this characteristic form, which is this 1 over f form, which is what we saw in that cartoon, that there's ever more mutations at ever lower frequency. So more and more mutations at lower and lower frequency. And so this equation is linear in 1 over f, and it has this parameter, which is the gradient of this linear line, which is mu over beta, where mu is the mutation rate per cell division, and beta is the death rate. And so this is a testable prediction now. We have a mathematical form that says this is what the variant allele frequency histogram should look like under neutrality, and we can go and look for this in the data. And moreover, if it does look like this, we can measure this parameter mu over beta and get some measurement of the in vivo per cell mutation rate. And so that's what we went to do. So you've seen this histogram already. This is actually whole genome sequencing of a stomach cancer. It's 100x whole genome sequencing from this nice paper from a few years ago. And so this is the cumulative histograms of just the subclonal mutations. So just looking in this frequency range down here, plotted against 1 over f, and you can see it really is a straight line. It really is linear in 1 over f. And so we can conclude that this tumour is evolving in a pattern which is entirely consistent with neutral evolution. And so the gradient of this line tells us what this mutation rate is. So we have a way now to infer these biological parameters and measure something which is previously inaccessible. Normally we describe the mutation rate in cancer just by asking how many mutations are there in the cancer genome, dividing that by the size of the genome, and we get a mutation rate per megabase. But that's not the same thing as the mutation rate per cell division. And the gradient of this line through this mathematical model gives us that new insight. It tells us what the mutation rate is per cell division. And so across the stomach cancers, this is what we measure. These are the measured mutation rates in microsatellite stable stomach cancers and microsatellite unstable stomach cancers. You can see we measure an order of magnitude difference, which would be in line with the expectation. And actually, the mutation rates we measure are quite high. And this is actually because the mutation rate we measure is the mutation rate per division, which leads to 2 surviving lineages. So it's the mutation rate divided by the death rate, and we can't scale out those 2 parameters. So that maybe explains the high number that we measure. So of course, there's lots and lots of this sequencing data out there. And so what we did next was ask, well, how often is the sequencing data that we observe consistent with neutral evolution? And so we took the data from the TCGA and asked how often does the allele frequency distribution follow this 1 over f-like distribution? And the answer was surprisingly often. So this is these are box plots of different tumour types with an R-squared goodness of fit to our linear regression model. And you can see that often more often in some tumour types than others, the data follows very well this 1/f-like distribution. In fact, in about 30% of tumours, the data is entirely consistent with this 1/f distribution. So it seems that this signature of neutral evolution is surprisingly common across tumour types. And I think this shows that maybe we've been very obsessed by looking at selection in the cancer genome, but not given due attention to the forces of neutral evolution, of randomness that also shape those cancer genomes too. So some of these cancer types are more neutral than others, and it's striking that cancer types such as colon and stomach, which infrequently seem to have subclonal driver mutations, tend to be neutral too, much as Christina just explained a moment ago. And so on the right, these are the measured mutation rates across different tumour types, and there's some sort of sanity checks in here. So things like lung cancer, where we might expect a higher mutation rate because of smoking exposure, do indeed have a measured higher mutation rate. Same for melanoma, where we might expect a high mutation rate because of UV exposure. So it seems to tie up. And so rather than this model of selective sweeps, what we see is something like this Big Bang model that Christina just explained. This Big Bang model seems to apply across many different tumour types, and we can see a good fit to this kind of model of neutrality in about 30% of cancers. And so in the Big Bang model, the predictions of our mathematical model are that the frequency of mutation is determined primarily by the time at which that mutation occurs rather than selection for that mutation, and that this seems to give this characteristic pattern that we see frequently across different tumour types. Okay, so that's great. So in about 30% of cases, we don't see any interesting evolution going on. What about the rest of them? What can we learn in that majority of tumours that don't seem to fit to this 1/f-like neutral model? The question is then, well, how is selection manifested in the genome, and can we measure the underlying evolutionary parameters that lead to those selection events that we see in the cancer genome? So again, our approach is to try and simulate cancer evolution under selection and fit these models to the data and measure these underlying evolutionary parameters. And so to simulate clonal selection, we do something like this. So in our neutral case, we have these series of cell doublings where the population grows over time. And if we have a new mutation in the neutral case, it's just a label that marks one of these lineages, as we discussed earlier. But in the neutral case, in the non-neutral case, again, we have this series of cell doublings over time with all these neutral passenger mutations accruing each round of cell division. But we model selection by one of the cells getting a particular alteration, say. It may be a mutation, it might be an epigenetic change, it might just be that that cell finds itself in a favourable microenvironment. But either way, the result of this selection on this particular individual cell is to cause the descendants of that cell, that particular lineage, to become overrepresented in the tumour compared to the neutral case. So the clonally selected lineage grows too much, in other words. And so it doesn't matter what the cause of selection is, ultimately the end result is just that the selected lineage becomes overrepresented in the tumour. And so we can classify selection by this parameter 1 plus s, which is just the ratio of the growth rate of the selected lineage to the rest of the cells in the tumour population. And what we're interested in is trying to measure this parameter 1 plus s in primary human tumours. And so to do this, we have this model, which is a stochastic branching process model. That's how we do it, and we simulate it on the computer. And what we want to do is to try and match the predictions of this stochastic branching processing model to real data. And so to do that, within the model, we simulate sequencing data and all of the sources of noise that exist in sequencing data. So within our model, we grow a tumour, and then what we do is take a virtual biopsy from our tumour where we take a sample of cells in that tumour. We do a virtual DNA digest where we just virtually extract all the alleles from the cells, so we throw away the cellular information. Then we take a sample of those alleles, which are effectively our sequencing reads. So we maybe sample 1 million alleles from the tumour, but actually only look at 100 of them, as we might do in a real sequencing experiment. And then we can transform these virtual sequencing reads into the variant allele frequency histograms that we obtain from real tumours. And so this is an output of the model, and you can see it looks very much like the data we saw earlier from real tumours, where we have this clonal peak of mutations and a subclonal tail of mutations in this case. Okay. And so, well, what does selection look like then? So we have a neutrally growing tumour where all of the lineages contribute equally to the future. And in this case, we have a tumour that's experiencing subclonal selection where one of the lineages contributes more than the others to the tumour. So what does this look like in the site frequency spectrum in these variant allele frequency histograms? So you've seen already that the neutral case gives you our null 1 over f distribution with ever more mutations at ever lower frequency. And the effect of selection is to give an extra peak in this distribution. And so this peak is visible because it's all of the mutations, all of the neutral passenger mutations that were accrued in this lineage before this lineage was selected, that then as this lineage grows are pulled to higher and higher frequency in the tumour. Those mutations become overrepresented in the tumour. So this peak is visible not because of the, say, driver mutation which causes this subclone to grow, but because of all of the passenger mutations in that clone which reveal it in the site frequency spectrum. And so critically here, it's the passenger mutations that are revealing the evolutionary dynamics, not the drivers themselves. And as I say, you see also in this selected case, we still have this 1 over f-like tail. And this is the mutations, the passenger mutations that are always occurring as cells divide and mutate their genome, both within the non-selected blue cells and also in the selected lineage. And you can see that because there are now 2 overlapping 1 over f distributions within this growing population, this distribution isn't strictly 1 over f anymore, but it's something similar. Okay, and so if we want to measure selection, then we can just compare this, the observed distribution where we think there's selection to the theoretical null distribution of neutrality, as Christina was explaining a moment ago. And this comparison lets us quantify the parameters which cause selection in this tumour. Okay, so the first thing we wanted to know was, well, what kind of selection might we hope to be able to detect from sequencing data? And the answer was kind of depressing and is not very much. So this is a heat map of the selective advantage of a subclone against the time at which that subclone appears. And the colour says when was there a detectable deviation from the neutral null model of the neutral null variant allele frequency histogram. And you can see that this heat map is only coloured in the central region of the plot. So when selection was either too weak or too late, we had no hope of seeing it because it didn't cause a significant deviation from the null variant allele frequency histogram. But interestingly, if selection is also very strong or very early, what happens is then there's a sweep through the tumour, and again we don't see selection anymore because then we're just looking at the neutral evolution that happens within that selected subclone. So there's only a small window of opportunity where we kind of catch a clone in the act of sweeping through, where we can hope to see subclonal selection in sequencing data. Okay, so the next thing we want to try and do is actually quantify what the selective advantage is of subclones that cause these additional peaks in the variant allele frequency histogram. So again, we do a little bit of maths and we can say, well, the number of mutations in a subclone, so integrating the total number of mutations in this hump, is a measure of when that subclone arose. So more mutations will accrue if the subclone arose later in a tumour's evolutionary history, and that's what this equation is saying. It's essentially just the mutation rate times the age of the clone with this scaling parameter of changing time into divisions. And then the size of the subclone is, of course, measured by its allele frequency in this plot. And so assuming exponential growth, we can write down how the size of the subclone is determined by the selective advantage of that subclone compared to the host population in the tumour. And both of these parameters are dependent on the mutation rate, which I showed you before we can measure from this 1 over f tail. So when we have those 3 bits of information, we can do the algebra and we get an expression for the selective advantage of a subclone from these other measurable parameters, which are the clone's frequency and its age, which we measure from the mutation burden in the subclone. And so now we have a handle, a mathematical model to measure selection in human tumours. And so to actually estimate S, we resort to doing approximate Bayesian computation, and we use approximate Bayesian computation so that we can represent all of the noise in the sequencing data, which is not captured by this analytic solution I just showed you. So I'm sure most of you are familiar with ABC, but just in case, it's a really cool statistical inference technique from population genetics. And what you do is sample some parameters from your prior that you're interested in estimating, simulate your model, in this case our model of tumour growth under selection, given those parameters. And the parameters we're interested in are the fitness advantage of the subclone, the mutation rate, and the time at which that selected subclone arises. And so the model produces some simulated data which we compare to the empirical data in our tumour that we've collected in the lab. And if the simulation matches the data, we accept our sample from our prior into our posterior distribution. And if it doesn't match, we throw that sample from our prior away. And then we just go around this circle many, many times, say 1 million, 10 million, 1 billion times, until we've built up an accurate prediction of an accurate representation of our posterior distribution. So if we use this technique in simulated tumours, this is a neutrally evolving tumour that we've simulated, and this is the VAF distribution that comes out of the simulation. The red line is the posterior distribution predicted of the VAF distribution in our simulation. And this is the posterior distribution for the number of selected subclones that are likely to exist in this tumour. And you can see the inference technique gets the answer right. Here it predicts that the maximum probability is for there being no selected subclones in this tumour. It's neutrally evolving. And so this is an example where there is one selected subclone. You can see this additional peak in the variant allele frequency distribution. And this is the posterior estimate of the number of subclones. And you can see again the inference technique gets the answer right. And importantly, it also measures the other parameters that we're interested in correctly too. So it measures the mutation rate, which is measured in this one. But interestingly, I think, and most importantly, it also measures the fitness advantage of that subclone with a high degree of accuracy. So this is a method to measure these subclonal fitness parameters in vivo in human tumours. So what we then did is go and look for very high-depth sequencing data where we can get accurate, where we can be suitably confident about what the VAF of these subclones really is, and apply our inference technique to those high-quality data and try and learn these evolutionary parameters about how strongly selected these subclones are. So this is some data from Andy Futrial's lab in lung cancer. So these are 4 regions of a single lung cancer, and you can see that they are all consistent with the pattern of neutral evolution. They all show a 1/f-like distribution of ever more mutations at ever lower frequency. And our posterior estimate for each of these regions is consistent with there being no selected subclone. And so these are 3 other tumours. So this is an AML case from WashU, which is whole genome sequencing to about 300x. It's some fantastic data set. And here you can see there's 2 subclones evident in this variant allele frequency distribution. This is a breast cancer case from the Sanger Institute in the UK, and again you can see there's 2 subclones here. And this is a 5th region of that lung cancer I just showed you with 4 neutrally evolving regions. This 5th region you can see has evidence of a subclone in it. And overlaid is these red fits of our inference method, and you can see that we can fit the data well. And so this is kind of the headline results. So these are the fitness advantages that we measure that those subclones I just showed you are experiencing within the tumour. And so the take-home message is that they're absolutely huge. We measure that these subclones have something like a 50% increase in their fitness, so they either divide twice as quickly, die half as much. And that's absolutely vast. If you compare that to the kind of fitness advantages that are observed in classical experimental evolution, they're normally of an order of just 1 or 2%. And we're seeing things almost an order of magnitude different. So we're seeing this potential for huge fitness advantages sometimes during cancer evolution. And so I find that kind of remarkable. And of course, these clones have to arise kind of early. We know that, otherwise we wouldn't be able to see them. So in general, it seems that the signature of selection is still quite rare, at least to the limits of the data that we have. So this is in our colon cohort when we apply our inference method. And the difference between what I showed you at the start of the talk is when we had an analytical form for what neutrality should look like. Now we have this stochastic simulation that takes into account all the sources of noise. When we have all those sources of noise accounted for, far more of the data that we have from colon cancers, this is TCGA data again, is consistent with this neutral model. The vast majority of colon tumours seem to be evolving neutrally, and a large proportion of stomach tumours too. And again, when we do see selection in these cases, we measure that the subclones have very large fitness advantages. So these numbers, I think, are really cool. They tell us something about the underlying evolutionary biology of the tumours, and that, we're able to quantify the evolutionary parameters of these tumours. But importantly, they allow us to make predictions about the future. If we know what the relative fitness of these clones are, we can both look back in time and say, well, what did the tumour look like previously, but we can also forecast forward in time and say, well, what will the tumour look like next week or next year if the selective environment stays the same. So that's what you're seeing here is just an example from this AML case where we've measured the fitness of these two subclones, and we estimate that this is the time in tumour history when we're making our samples. And so you can see there's one subclone that's predicted to die out effectively in the tumour, while the other subclone will come to dominate that tumour. And with these, this is just one toy example, but you can imagine with these kind of predictions, we might be able to think about how to tailor treatments better in order to maximize the efficacy of the treatment, given that we know how the tumour will change anyway, even if there was no treatment applied. So this is a basis for mechanistic prediction of forecasting prognosis in tumours. All right, so that's all I wanted to say. So I'll show you some data showing you how the cancer genome is dominated by the signal of tumour growth. And in fact, we see a signature of neutral tumour evolution very, very commonly across many different tumour types. And neutral evolution means that the frequency of subclonal mutations in a tumour is determined principally by the time at which the alteration occurs rather than any selection for that mutation thereafter. And I showed you in cases that don't conform to the model of neutral evolution, there are selected subclones sometimes, and those subclones are incredibly strongly selected. They have very, very large fitness advantages indeed. And so I just thank the collaborators, particularly Mark, who's done all the work in the lab, and Andrea, who we do all this work together with, and our funders, Cancer Research UK and Wellcome. Thank you very much.